starting off with dilution, the formula for it is volume added divided by total volume, and dilution equals D. And then the dilution factor formula is 1 over the dilution. And then the cumulative dilution factor is you're multiplying all of the dilution factors. So like 1 over 100 times 1 over 10 times 1 over 10. And then plate dilution factor is the CDF, the cumulative dilution factor, times the volume plated, and the number of CFUs per milliliter formula is colonies counted divided by plate dilution factor, and then the total CFUs in the sample formula is CFU per milliliters times the total original sample volume. And then to use the graph of absorbance, optical density versus concentration. Optical density is used to estimate cell number. More cells equals more turbidity, which equals more light absorbed, which equals higher OD. The equation y equals mx plus b is used on a straight line to convert the OD to cell concentration, and always use units like bacterial cells and scientific notation. So standard plate count advantages, it counts living cells only. Cheap equipment. Disadvantages are it's only viable for 30 to 300 and time consuming and only works on cultural bacteria. And indirect methods like spectrophotometry, NPN, etc. Advantage equals fast and requires less manual labor. Disadvantage only estimates total cell count, can differentiate between alive and dead cells. You can enumerate bacteria by direct methods like membrane filtration, Petroff, Hauser, cytometer, Coulter counter, and flow cytometer. And then indirect methods like turbidities, which is done with a spectrophotometer / OD, metabolic activity, which is minus waste product, which equals growth estimate, and dry weight. Direct methods count the actual bacteria, and then indirect methods count what the bacteria produce or affect. And a CFU is a colony forming unit. CFU per milliliters is used instead of bacteria per milliliters because the bacteria form in clumps. And for counting them, it shouldn't be, for CFUs and SPC, it shouldn't be less than 30 or greater than 300. And when it's a lot, you split it in half and count one side and multiply it by 2. And then for the dilution formula, we already went over those, but I'll go over them again. Dilution is volume added divided by total volume, and then dilution factor is 1 divided by the dilution. Cumulative dilution factor is to multiply all the dilution factors. Plate dilution factor is the cumulative dilution factor times the volume that's plated. And then CFU per milliliters is colonies counted divided by the plate dilution factor. And then the total number of CFUs in a sample is the number of CFUs per milliliter times the total original sample volume. And then the difference between cumulative and individual dilution is the cumulative one is the product of all the previous steps, including the one that you're on. And then individual dilution is dilution at one step. And then E. coli is a good choice for a microbe in an enumeration experiment because it's motile, single-celled, and it doesn't clump, so it's easy to culture. And then to make a line of bacteria, you use a sterile swab to spread it evenly across the agar, and the purpose is to measure how effective an antibiotic is across the whole surface. The zone of inhibition is the clear area around the antibiotic disc where bacteria didn't grow, and it's formed because antibiotic diffuses out of the disc into agar and kills/stops bacteria. And then the relationship between the zone of inhibition is if you have a bigger one, the antibiotic is more effective. If it's a smaller one, then it's less effective. And then antibiotics are chemicals that kill or inhibit bacterial growth. And then for the effectiveness of antibiotics, if it's sensitive, it has a larger ZOI. Intermediate, it has a medium ZOI. And if it's resistant, it has a small or no ZOI. And then you use the table to measure the ZOI in millimeters. And then the Kirby-Bauer method is when you have the Muller-Hinton agar 4 millimeter plate thickness and 0.5 McFarland bacteria concentration. And then broad spectrum antibiotics kill a wide range of bacteria and acts on both gram-positive and gram-negative bacteria. In narrow spectrum, it just targets specific bacteria types. Viruses are called obligate intracellular parasites because they can only replicate inside of a living host cell. A bacteriophage is a virus that only infects and replicates in bacteria. Bacteriophages only infect prokaryotes, and viruses aren't alive or dead. A virion is the complete virus particle that's outside of the host cell. A dormant virus in the virus cell is the host cell that's actively being infected or hijacked by the virus. And viruses use specific surface receptors to attach like a lock and a key. And then PFU stands for plaque-forming units. Phage plaque is the clear zone or bacteria line where bacteriophages kill the bacteria. And to calculate PFU per milliliters, it's the number of plaques that you counted divided by the dilution factor times volume plated. So the number of plaques is at the top, and then the dilution factor times the volume plated will be at the bottom. And then a countable plate is for bacteriophages is 25 to 250 plaques. And then again, the SPC is 30 to 300, and then the bacteriophage plaque one is 25 to 250 because the phages make bigger, blurrier zones. And then the purpose of the bacteriophage lab was to calculate concentration of bacteriophages in a sample and quantify how many PFUs are in a sample. And then The SVC lab and PFU lab are the same because they both use serial dilutions and they're on agar and you want a countable plate, and it uses concentrations per milliliter. And the differences are because SVC counts bacteria, phage titer counts viral plaques, SVC measures bacterial viability, and phage titer measures viral infectivity. And then the SVC count range is 30 to 300, and the phage titer count range is 25 to 250, and the phage plaques overlap and spread differently compared to heat colonies. And then the 3 outcomes / cycles of an infection by a bacteriophage is lytic, and that's the immediate destruction of the host. Lysogenic, which is when the virus is hiding in the host DNA and then multiply. And then chronic is when the virus is present but non-active, but still shedding the virions without cell death. And then the phenol red is the pH indicator that was used for the unknown bacterial one. And it's used in tests like red broth sugar fermentation, like a glucose or lactose test, and Durham tube test, which is for the gas production. In stem media, the S is for sulfur reduction, the I is for indole reduction, and the M is for motility. And for a positive sulfur reduction, that means that you have black precipitate in the tube, which shows the H2S production, and the negative is obviously no black precipitate. And then the indole production, the positive one, it has like a red ring on the top of the whatever it is after adding Kovacs reagent, and then negative is no red ring. Motility, positive, it's like it looks fuzzy / cloudy all over the tube, but not just on the stab line. And if it's negative for motility, that means that the bacteria don't move from except from where you stabbed them in the agar. And then the thioglycolate, obligate aerobes, for the oxygen gradient, they grow at the top of it only, like where that pink part is. And then the facultative anaerobes, they grow all throughout of it, but more at the top. And then obligate anaerobes grow at the bottom only. And then you shouldn't shake the thioglycolate medium because it has an oxygen gradient, and O2 is rich in the area at the top. And then if you shake it, then it's fucked up because it can't determine the oxygen requirements anymore. And the API 20E strip, it can only be used with a pure culture because mixed cultures would have confusing results because different bacteria would have different reactions to it. And the advantages of this type of test is that many tests are done simultaneously and they're quick and easy to use, but disadvantages are that they're expensive and some fastidious microorganisms might not grow on it. And yeah, and then you just do the positive and negative results and then be able to see what it would be like on the flow chart. And then wells are over or underfilled to control the oxygen availability for specific tests. So if you underfill it, that means that the test needs to occur in aerobic conditions. And if you overfill it, that means it needs to occur in anaerobic conditions. And then bacterial transformation, the 3 types of gene transfer are transformation, which is when it takes naked DNA from the environment, transduction, which is the transfer of DNA via a virus, and then conjugation, which is a direct transfer of DNA through the pilus between bacterial cells, like when it sticks onto the other one. And then artificial gene transfer is useful for producing medicine and genetically engineered crops. And then transformation is the uptake of foreign DNA from environment into bacterial cell. Competence is the ability of a cell to naturally take up foreign DNA. Artificial competencies is when it's a lab-induced ability to take up DNA and it increases the frequency of transformation. And genetic engineering is the deliberate modification of an organism's genetic material using biotech. And inducible operons are DNA unit that's switched on in response to the addition of a metabolite that regulates the operon. And then repressible operons are ones that are usually turned off. And then whenever it has enough of the specific metabolite. And a plasmid is a circular small DNA molecule separate from chromosomal DNA, and it often carries beneficial genes. And genes are regulated by operants, which can be turned on or off based on environmental signals. And 2 methods for artificial transformation is the heat shock method, which is a sudden temperature change that makes bacterial membranes permeable to DNA. And then electroporation, which is when electrical pulses create temporary pores in cell membranes, letting the DNA in. And then for the heat shock transformation, you need chemically competent bacterial cells prepared with CCl2, you need plasmid DNA, ice baths, and a heat bath. And then the role of heat in ice bath, the ice bath prepares bacteria, stabilizes cell membranes, and holds bacteria in receptive state. And the heat shock temporarily disables membranes and allows DNA to slip into the bacterial cell. And then the plates that should have growth are the ones with the LB and ampicillin with the positive plasmid, and the LB amp ara with the positive plasmid, and the LB with the negative plasmid. And then no growth should be the LB and the amp with the negative plasmid because it has no resistance, so the ampicillin has the ability to kill it. And then the controls are for positive, it's the LB with no antibiotic, and then it just shows the bacteria is alive and capable of growth. And then the negative control is the LB amp with no plasmid. It shows amp kills non-plasmid cells. And then the selective agent is ampicillin, which ensures that only bacteria with the plasmid will survive. And then the regulatory or inducible factor, it's actually inducible and it's arabinose, and then it's induced by arabinose sugar, which switches on to produce the green fluorescent protein. And then if the transformation wasn't successful, like if the plasmid didn't go through to the bacteria, then the LB AMP plate with the positive plasmid would have no growth because it doesn't have any resistance because it didn't pick it up. And then the LB AMP RO would also have no growth because no resistance. And then the LB with no plasmid has the regular growth because it doesn't have any ampicillin on it to kill it. And then the BLA gene is on the PGLL plasmid, and it encodes the beta-lactamase protein and provides the resistance to ampicillin, and it's always on. And the GFP gene is what encodes the green fluorescent protein, which causes the green glow by UV light. And the ARC gene, it encodes the C protein, which is regulatory and controls expression of GAP, depending on arabinose presence. And then in the experiment, it's the inducible operon. And the GFP gene is only expressed when arabinose is present, and without arabinose, there's no glow. And then the sugar that's used to control the ARC gene regulation is arabinose. The only plate that should glow is the LB AMP RA with the positive plasmid because it has the arabinose sugar and the plasmid, and then the rest wouldn't glow because the LB AMP with the positive plasmid has no arabinose, so it wouldn't glow. And then the minus for the plasmids don't grow because they don't even have the plasmid, so they don't have the GFP gene. And then 4 agar plates were used because we need to check for normal bacteria growth, which is in the LB without the AMP, and then the effectiveness of ampicillin, which is the LB with AMP, and then the transformation success without arabinose, which is the LB AMP with the plasmid, and then the transformation plus the success with the GFP, which will be the LB AMP auto. And then the objectives of this lab, YAP. And then the purpose of the bacteriological examination of water was to detect the presence of coliform in the water, and coliforms are indicator organisms used to assess whether water is contaminated with fecal matter and possibly pathogenic microorganisms. And the MPN, which is most probable number, is out of 100 milliliters of water, and then it's estimating bacteria concentration based on positive/negative results. And then we test the water for contamination because if the water is contaminated with shit, then it can carry diseases causing organisms like Salmonella, Shigella, and pathogenic strains of E. coli. And then detecting contamination helps prevent waterborne outbreaks. The 5 characteristics of a coliform bacteria, so think of FAR, flag. So FAR, facultative anaerobe, and it's rod-shaped, and then it ferments lactose to produce acid and gas, and it's negative for spores, so it doesn't form spores, and it's a gram-negative bacteria. And then the 5 characteristics of a good fecal indicator organism is that it normally is only in the intestines of mammals and birds, always found in fecal matter, survive outside of the body for a limited amount of time since they're non-spore forming, relatively easy to identify, and not normally present in soil or water. And then the pH indicator for this one was phenol red, and the sugar was lactose, and the confirmed test agars are EMB and then the endo. And EMB stands for eosin methylene blue. And then all the tests that were used, presumptive is the lactose fermentation in the Durham, which are the DFT tubes, confirmatory, which is the growth and colony appearance on EMB or endo agar, and then the completed one is the gram stain from colonies and lactose fermentation confirmation. And then you can't test for every specific pathogen because it's expensive, and E. coli is a representative indicator because its presence suggests fecal contamination and possible presence of pathogens. And then in the EMB, it's selective and differential. It's selective because it inhibits gram-positive organisms, and then it's differential because it does lactose fermenters versus non-fermenters. And then E. coli appearance is dark purple colonies with green metallic sheen. And then the endoplate is selective and differential because it inhibits gram-positive organisms. And then the same thing, it does lactose fermenters, but it turns colonies red with a metallic sheen, and then like a gold-green metallic sheen. And then the EMB agar, like the original color, is dark purple blue, and then the endo color is hot pink. And then the DVT tubes are a culture tube with a small inverted vial, which is called a Durham tube, to trap gas produced during fermentation, which is a silvery-looking bubble. And then the positive DFT is when you have the yellow color, which represents acid production and a gas bubble inside of the Durham tube. And a positive presumptive MPN only suggests possible contamination, so some microorganisms other than coliforms produce acid and gas from lactose fermentation. And also the order for the Durham tubes is the 10 milliliter to 1 milliliter to 0.1 milliliter. And then I and VIC test. The I is the indol test from the sim tube, M is the methyl red test, V is the Vos-Prescour test, whatever that means, butanediol fermentation, and then C is the citrate utilization. And then SBA is enrichment and differential. MSA, endo, and EMB are selective and differential. And the SP plate color looks cloudy red, and it's whatever colony color, and then it has no sheen, but it has hemolysis. So the beta BCC is complete clear, so it's complete hemolysis, so the plate won't look cloudy anymore, it'll look clear. And then alpha partial hemolysis, that's like when it turns like a greenish kind of color or brownish color, and it's still kind of cloudy, but it's not like completely lysed, the bacteria. And then, the blood. And then gamma is no hemolysis, so it's just cloudy and there's nothing around it. And then the MSA plate color is the orangey red clear one, and then the colony colors are red or yellow and there's no sheen, and that's the one where it turns yellow if there's acid. And then endo plate color is the clear hot pink, and the colony colors are either pink or red, and it has a green metallic sheen. And then the EMB plate color is the deep purple/blue, and then the colony colors are purple with a green metallic sheen if it is the coliform. And then for the medical microbiology lab, we use MSA, mannitol salt agar, and SBA, which is sheep blood agar. And then the property of mannitol salt agar that makes it selective is the high salt concentration. So Staphylococcus like the salt. And then staph can grow on MSA, strep cannot grow on MSA. And then Staph aureus changes the color of the MSA plate to yellow because it ferments the mannitol sugar, which produces acid and turns the pH indicator in agar yellow. And the carrier state for Staph A means that you have it on your skin, nose, gut, or mouth and are asymptomatic, and it's usually harmless, but some strains can be pathogenic. And then sheep's blood agar is enrichment because it supports the growth of fastidious organisms by providing additional nutrients, which is the blood. And it's differential because it shows hemolysis patterns, which is the gamma, beta, alpha, and it differentiates bacteria based on their ability to lyse red blood cells. The pathogens that's found in the nose and throat, just remember Staph A, it has yellow colonies and it does completely clear hemolysis. And then a candle jar is a sealable, non-flammable jar large enough to hold petri dishes, and then you use it to get low O2 in it, but then a high CO2, and then it allows for the growth of facultative anaerobes or microaerophilic, that means. And then hemolysis patterns, the alpha is green, partial red blood cell lysis, and examples are strep mitis and strep pneumoniae. Beta is the clear zone complete lysis, and that's strep pyogenes and Staph A. And then gamma is no color change, no hemolysis, an example is Staph epidermidis. And then staph are in clusters and they grow in the MSA, and they have yellow medium large colonies. And then strep are chains and they don't grow in MSA, and they're small translucent colonies. Electrophoresis is the process of separating DNA fragments based on their charge and size by applying an electric current through the gel matrix, and it works by negatively charged DNA molecules moving through agarose gel when an electric current is applied, and the smaller fragments migrate farther and faster because they can move easier through the gel's pores. And then for all the parts is the gel tray, the agarose gel, the gel box chamber, the power supply, the cone, which is to create the wells, and the buffer solution. And then the charge from DNA is negative because of the phosphate backbone, and then DNA moves to the positive electrode, which is the red one. And then the smaller ones move faster, the larger DNA fragments move slower. And then to determine the size of a PCR fragment, you compare migration, so the longer and larger fragments will be closer to the wells because they can't move as easy, and the smaller, shorter ones are farther away. And then the kind of electrophoresis that DNA sequencing uses is capillary electrophoresis, which is from small to large, and then whatever that means. And then you would diagnose patients using PCR and gel electrophoresis because PCR is used to amplify specific DNA sequences from patient samples that may contain a pathogen. And after amplification, the gel electrophoresis is used to separate DNA by size. And if bands appear at the expected size for the pathogen, then there's a positive result. And nucleic acid tests are detect the viral or bacterial genetic material. And then nucleic acid amplification tests are when they amplify tiny amounts of genetic material to detectable levels using PCR. And then PCR is polymerase chain reaction, and it amplifies specific DNA sequences exponentially. And in cDNA, you have to have the nucleotides, the buffer, the primers, and the polymerase. And then it's heated and cooled, then denaturation, then annealing, and then extension, and then heating and cooling repeated. And then a PCR fragment is made of DNA fragments produced by amplifying a specific target region between two primers. And a primer is a short single-stranded DNA sequence that binds to the target DNA and provides the starting point for the DNA polymerase to begin extension. And then RT-PCR is reverse transcriptase polymerase chain reaction. And then reverse transcription is when the viral RNA is converted into cDNA by using the enzyme reverse transcriptase. And in SARS-CoV-2, the viruses are collected using nasal or throat swabs. And then for the majority of nucleic acid tests used for SARS-CoV is RT-PCR, and then COVID is transmitted through infected respiratory droplets, and it has similar symptoms to the flu, and it's worse than the flu because it has a longer incubation time and higher hospitalization rates, and it affects the respiratory and circulatory and digestive and nervous systems and your kidneys. And then a zoonotic disease is a disease transferred from animals to humans. And then The gram stain, a positive one is purple, and a negative one is pink and red. And then endospores, the actual endospore is green, and the vegetative cells are red. And then yeah. The individual dilution is the volume of the liquid that you added to the tube divided by the total volume of liquid in the tube. So, bottle A originally has 99 milliliters of water and 1 milliliter of sample is added. So you do 1 milliliter, which is the one that you added, and then 99 plus 1, which is 100, which is the total volume in the tube. And then you divide it and you get what you get. And the cumulative dilution is like how... it's the product of each individual dilution. So you get 1/10, 1/10, and so on time, multiply it together.